the product can be RNA itself, or finally it can be a protein. What I'm just trying to emphasize is that it's important for us to understand that whether this process of transcription is taking place on a secular chromosome, such as in bacteria or prokaryotes, or whether this transcription is taking place on our chromosomes, which are linear, one thing in common is that we are all carrying genes. Of course, the number of genes differs, but the principle here is that the process of transcription is not retrieving all the information that is carried on these chromosomes. It's retrieving information that is required by the cell. And it makes a lot of sense. You can imagine you and me right now, we don't make our own energy. We have to, eat food, and then the body can get energy from there. So the cell or the body itself has found mechanisms to conserve energy. Remember, this process is energy dependent. Even replication itself is energy dependent. I think we saw that. So you just don't want to be expressing genes anyhow. You can imagine a baby who's born today, like I always say, you will never find a baby born with, boobs and all other stuff that we have at adolescence. This is because maybe when they are born, certain genes are not expressed, or should I say as much as they should, but they have that potential. And as they grow, of course, features are going to indicate to the system to say, I think now you must transition into puberty. And at that point, there will be gene expression for the relevant hormones to be expressed in the quantities that are needed. So before that time, the body should not spend any energy expressing genes that are not required. So think of this process not just by the understanding of a gene as a segment on a chromosome, no, but you must think of it broadly, even its implications in terms of energy conservation. And also to realize in all these systems, whether it is a multicellular organism such as yourself and me, or a simple organism such as a bacteria with only one cell, we all have to conserve energy. And one way is to be able to transcribe genes as and when we need them. Now, I also want to look at the structure of the genes. At this point, we've compared transcription site in prokaryotes and in eukaryotes. We've also looked at understanding that the cell is only retrieving a segment of DNA to transcribe it into RNA. At this point, I want us to look at the structure of these genes. So what you see here on this is a segment, a gene from a bacteria, a prokaryotic cell, and this one here is a gene from a eukaryotic cell. Now, keep in mind, I've retrieved only a segment, meaning that on this end, there's more genes, on that end, there's more gene. Remember, this was a loop. And in our eukaryotic cell, ours is linear, isn't it? But of course we know this gene is coming from a linear chromosome. What this means is this way, there are other genes, and of course, maybe this side, there are also other genes. It could also be the end of the gene, the chromosome. It could also be the beginning of the chromosome. This is the first gene. But I want you to understand, what you're seeing in this slide is just a segment I've pulled out from a chromosome, either secular or linear. Now, here we want to look at the structure or the organization of these genes. One thing we're going to note is that in bacteria cells, their genes are uninterrupted. They're kind of continuous. That is to say, after this gene, if this was gene 1, the next thing is going to be gene 2, gene 3, and so on and so forth. They're uninterrupted. While in our genes, we find that within the gene, there are interruptions. This information that is being transcribed into RNA, it has got interruptions in it. The information is not much. The information still requires to be processed for it to make, something meaningful. In short, what I'm trying to say here is that if you look at this gene, you find that the information is continuous. It doesn't need any further, processing. The cell can make sense out of it. In short, the genes for prokaryotic cells do not contain introns. Introns, these are segments of DNA that are found within the gene that do not carry information for making a mature RNA. And by mature RNA, I mean functional RNA or a functional protein. Sometimes they're also referred to as junk DNA. When you think of junks, or should I say, when you think of junk material, it's information that we don't need. It is not really necessary. Of course, we know that these introns, they're important. They even help in the way this material is processed. But the understanding here is that this information in this gene is continuous. The cell can make a functional RNA molecule. And by function, meaning it can function to serve a particular function in the cell. But our genes are interrupted by these introns. In this slide, they are shown in yellow. Now, these introns, think of them as junk DNA. So meaning that an intron is found within the gene, in this case, as you can see in this slide, and this intron is not carrying information that will finally end up in the mature functional RNA, or if it is a protein, it will be removed because it is carrying junk information. It is not really needed for a functional RNA or a functional protein. Now, it means that the regions of the gene that carry information that finally ends up into a functional RNA, these regions are referred to as exons. What this means is that in eukaryotic cells, our genes have exons. Exons, these are the coding regions, or should I say, these are the portion or segments of the gene that carry information that will finally contribute to making a functional mRNA molecule. In prokaryotes, their genes are continuous. They do not have introns interrupting the mRNA. Sorry, the DNA. What I'm trying to say here is that when you look at the gene organization, it's important for us to understand that in prokaryotes, the genes, the segment of DNA that is going to make the mRNA molecule is not interrupted. It is continuous information. What continuous information means is that the product is going to contribute in the way it is to a functional RNA molecule or a protein. In eukaryotes, our genes have both exons, which are the coding sequences, or should I say, segments or sequences that carry information that will contribute to a functional mRNA protein, while the introns, these are junk DNA. This junk DNA does not contain instructions that are going to be part of the mature or functional mRNA or the protein. And because of this, it means that when the mRNA molecule is made from a bacteria genome, it can straight away go into making a protein. While for us, when an mRNA molecule has been transcribed, it will carry some of this junk DNA. Because it carries junk DNA, which should not be part of the functional mRNA molecule, this material needs to undergo processing. That is to say, the mRNA that is formed in our eukaryotes. And that is why at the beginning, I mentioned some of the things we're going to look at is processing of the mRNA. Why is this mRNA being processed? Because of the nature of the genome organization. Ours, that is yours and mine, our genes are interrupted by introns. Introns, they carry essential information, but not for making the mRNA molecule. This information is used for sometimes controlling how this the mixing of these exons. We're going to look at how we can get more than one protein from a gene. Sometimes they can help in that regulation. They can also help regulate the efficiency with which this RNA is formed. So when I say junk, I'm not trying to say they're useless. No, they're like spacers. They have got very important functions. Of course, initially they were just thought to be useless, but this time around with research, we know they do play a role. Sometimes they play a role in the way these exons are joined, and sometimes they also help in regulating the efficiency of transcription. So there are many functions, but we are looking at them as junk DNA from a point of making a functional mRNA molecule. So we have some notes that you can, in your own time again, you can read and understand what I was trying to say. more comprehensively. So at this point, I want us to move on to the DNA. I think one of the questions. DNA is a double-stranded molecule. But the material that we're making, in this case, through transcription, the RNA, is single-stranded. So now, are both strands of DNA going to be used? Or only one of them is going to be used as a template? The question we're trying to find out here, are both strands of DNA used in transcription as templates, or only one of them serves as a template? So this question, we can look at this slide. So what we see here is one is a double-stranded DNA molecule. We have got a strand in red moving from the 5' to the 3'. We have the strand in blue moving from the 3' to the 5'. And we know why this is designated as such, because we know DNA has an anti-parallel nature. One strand's moving the 5 to 3, the other one in the 3 to 5. And what you see here in purple is the RNA that is being formed through the process of transcription. Now, are both strands of DNA used in transcription? No. Only one strand of DNA is used in transcription. That is to say, only one strand of DNA is serving as a template. And at this point, we know what a template is. Like I always say, if you're a company printing T-shirts with maybe an I love you sign, you need to have a template with a love sign, because if you don't have that template, it means every time someone orders, you have to go and recreate that. But normally, you are going to have a template, like a pattern, that you can just go and retrieve and quickly start making or printing those T-shirts. So when you think of a template, in this case, we are looking at the strand of DNA that is going to be used to form complementary base pairs for the formation of our mRNA. Remember, we are saying RNA, but we know this should be messenger RNA, right? Because we said there are those 3 major RNAs. So this one here is messenger RNA. So the strand of DNA that serves as the template for synthesizing our RNA is referred to as the template strand. Sometimes it is also referred to as the antisense strand. The strand of DNA, because there are 2, that serves as the template. That is to say, complementary base pairing is going to take place with our strand, is referred to as a template strand, sometimes referred to as the antisense strand, sometimes it is referred to as the non-coding strand. It's got so many names, okay? And the other strand of DNA, this one in red, the one that is not acting as a template strand, is referred to as the sense strand. There's a reason why it is referred to as the sense strand, and I'm going to use a slide to just demonstrate that. But for now, we just want to understand to say, while DNA is double-stranded, during transcription, not both strands are used as a template. Only one of them is used. The strand of DNA that is not used as a template strand is referred to as the sense strand. It is also referred to as the coding strand, sometimes also as the non-template strand. Now, let's look at the orientation of the template strand. The template strand is a strand that is in blue. From this end, it is a strand moving from the 3' to the 5'. The strand that is serving as a template from this end, and the assumption here is that transcription is taking place from this end where my cursor is to this end, is a strand that is moving from the 3' to the 5'. What this means is that the enzyme that is synthesizing our RNA is reading the strand, that is getting this information from the strand by moving from the 3 to the 5'. But as we go on, we realize that synthesis is always from the 5 to the 3 prime. If I were to label this strand to designate which one is a 5 to 3, this end of this RNA should be the 5 prime. And this end of the RNA should be the 3 prime. What I'm trying to say here is that the enzyme, we are going to note in the slides that follow in this lecture, that the RNA polymerase, which is the enzyme that synthesizes RNA, reads template strand from the 3 prime to the 5 prime. That is reading. But synthesizing is from the 5 to 3. And during DNA replication, I made it very clear what the 5 to 3 prime is. And I remember very well to say, I urged you guys, you go and look at the structure of the nucleotide. Okay, look at where the 3 prime is sitting and where the 5 prime end is, looking at the numbering of the pentose sugar. So at this point, we're very clear. If you don't understand, I think we can still cover that with some that don't understand, but the first thing you need to do is just understand the structure of your nucleotide. How we label the 5 and 3 and look at the process of transcription, in this case to say the polymerase will read the template strand from the 3 to 5, but synthesis here is 5 to 3. In addition to that, before I move on to RNA, of course, I'll mention it again. RNA also carries ribonucleotides. And during this synthesis, if on the template strand you had an adenine, here we should add a thymine. But in RNA, we do not have thymine. In its place, we have uracil. So just keep that in mind. So at this point, it's just trying to kind of emphasize this question to say, how is the coding strand? The coding strand is a non-template strand, and the template strand, how are they related during transcription? We just want to see if we can just look at them, and understand why one is a coding and why one is a template strand. And here, I want to emphasize this because if at all I were asked to determine the structure of my RNA, can I just pick one of the 2 strands and that will be the correct strand of the mRNA to be made? Let's look at that. So right now we have the strand in green, and this one we're saying is a non-template strand. It can also be referred to as a sense strand, coding strand. They all mean the same thing. And this one here is the template strand, antisense, non-coding, or the strand that is being transcribed. So meaning that complementary base pairing would take place with this one, or the RNA polymerase, the enzyme that is going to help us synthesize our mRNA, is going to be reading this strand from this direction to that direction. Now, and then the mRNA that is going to be synthesized, and at this point we are assuming that transcription has taken place, is this RNA. I want us to compare the nucleotides in our product to the green strand. When you're done with that, I want you also to compare the nucleotides of our product to the template strand. So let's start with the green strand. At this position, we have adenine. On our mRNA, we also have adenine. The next position is the thymine. The next position here is a uracil. But we know uracil and thymine, if this were DNA, this would have also been thymine, but because this is RNA, we have uracil. The next position is guanine. And at this position also we have guanine. So as you go along this strand of the RNA that has been produced through the process of transcription, you're going to find that the newly formed RNA has got the same nucleotide combination as the coding strand. The only difference is that where there is a thymine in the coding strand, in your RNA, there will be a uracil. Let's go on to this other strand. In addition to that, one thing you're also going to observe is that the direction of RNA is the same orientation as the coding strand. This end was a 5 prime, this end is a 3 prime. And everything else that follows from the 5 to 3 is the same, apart from where we do not have thymine in RNA. In fact, if I'm given a set of nucleotides and I'm asked to pick which one of them are RNA origin or DNA, if the strand has got uracil, I know this must be RNA. If the thymine, I'll know this is DNA. Now, let's go back to our strands. We're now looking at the strand. If you start from here, you can see this is a thymine. If we go to our RNA product, this is adenine. Now, this is because we know that thymine complementary base pairs with adenine. On the next position, we have adenine. Adenine complementary base pairs with thymine. But because this is RNA, it complementary base pairs with uracil. We can go to this other end. We have adenine complementary base pairs with uracil. So what I'm trying to say here is that when you move along the non-coding strand and you also move along your mRNA, you can see there is complementary base pairing. One thing we note here is that transcription follows the rules of complementary base pairing. It is not breaking any rules. The only thing we're going to see is that there will be addition of uracil instead of thymine because now we're making RNA. In addition to that, we're also going to see, or maybe just make a reason why this is the antisense and why this strand is called the sense strand. Because the sense strand is the exact copy of your newly formed mRNA, apart from the fact that it will have uracil and not thymine. So the name sense is because of this being an almost exact copy. Now keep in mind, that is not to say that the information in this mRNA is different from this information that is being carried in the template strand. No, the information is the same. Remember, this is complementary base paired with the coding strand. So the information that is in here and there is the same. But here it's trying to distinguish that DNA is a double-stranded molecule, but only one of those is being used as a template, meaning that both of them carry the same exact information, but only one of them is serving as the template in this process. That is to say, if you are asked to predict the structure of the nucleotide combination of your RNA, I don't have to start, complementary, writing them out or figuring them out one by one. No, the first thing that I need to do to establish is first the orientation of the slides of the strand. What do I mean by that? Check which one is the 5 prime, check which one is the 3 prime. We know that the RNA polymerase must read the strand from the 3 to the 5 because synthesis is in the 5 to 3 prime direction. Then I'll know this is my template strand. If this is my template strand, this is going to be my non-template strand or the coding strand, meaning that my mRNA will be the exact copy of this coding strand. I'll just replace the T with a U. There's no need for us to memorize, it's just to understand what is going on here. And of course, you have notes and more notes. These 2 slides are for you to just go and look at, and of course, this is a summary. It is still the same thing. Nothing really here has changed. What I was trying to say here is that you have your DNA acting as a template, the base sequence of the coding strand, the base sequence of the mRNA strand. You're going to find that the coding strand and the mRNA transcript are going to have the same nucleotide combination. Just remember, these are deoxynucleotides. These are ribonucleotides, and we know why that is so. The pentose sugar, the nucleotides that we are using to make DNA, are different from the sugars that we're using here. This is a deoxyribose sugar. So that is also one of the differences here. I think it's important for us to carry on that information. Okay. So the next thing we're going to look at now, with the understanding of what transcription is, we've understood what the products of transcription are, we've also understood where it is taking place. and also that DNA is a double-stranded molecule, but our product is one, so meaning that only one of them serves as a template. We want to find out, how is this process taking place? Who is, or what is the enzyme that is responsible for transcribing it? That is to say, similar to DNA replication, it is an enzyme-dependent process. Transcription also requires enzymes. The enzyme that is responsible for transcribing our RNA, for making our RNA, is RNA polymerase. And I've mentioned to you to say, the RNA polymerase, it reads. And what I mean by reading is that it will start getting its information from this template strand, starting from the 3' direction. That is where it's going to start reading. Okay? And it's going to, meaning the RNA polymerase is moving on the template strand in the 3 to 5' direction, while the synthesis of the complementary RNA molecule is 5 to 3' direction. And also, I mentioned to say, the rules of complementary base pairing are also followed during the process of transcription. But we need to keep in mind that in RNA, we don't have thymine. We have uracil. In addition to that, these nucleotides, they are ribonucleotides because the pentose sugar there is a ribose and not a deoxyribose. Just some things that we need to take care of. In addition to that, the process of transcription needs to start from somewhere. That is to say, on the gene, there must be an indicator to say that this is where the gene starts and this is where the gene ends. One of those markers or signals that guide transcription to ensure that we get our transcript from the beginning to the end, at the beginning, is the promoter sequence. I'll look at it in the next or so slides. I'll explain that later. But for me on this slide, is just to understand that we understand what a gene means. The gene should have a beginning where it starts. It must also have an end where the instructions end. So the gene should have certain features that allow transcription to know that this is where I should start. And one of those sequences is what we're referring to as a promoter. And it must all have a position that tells the process of transcription to say this is where this information ends. That region is the termination site. But we're going to look at them as we proceed. So let's look at the enzyme, the RNA polymerase. We're saying that RNA polymerase, this is the enzyme that synthesizes our RNA. This enzyme, like DNA polymerase, also synthesizes DNA from the 5 to 3' direction. Now, remember, when we're looking at DNA polymerase during replication, DNA polymerase, I used not a very good word. I said it was a DAO enzyme because it cannot initiate replication on its own. It needed a primer. And remember, we said the primer that we needed was RNA primase, or should I say just primase? It's an RNA polymerase. Now, the question you want to ask yourself here is that during transcription, is this RNA polymerase also a DAO enzyme? Does it also need a primer to initiate synthesis? No. RNA polymerase does not need a primer to initiate transcription. It can start transcription without the need of a primer. And remember, a primer, when we looked at DNA replication, is just a short segment of RNA. It was RNA in nature, so that's why it removed at the end of the day. That is synthesized by an RNA polymerase, the primase. But the RNA polymerase is a bit smarter. It can do it. Of course, we're going to see as we move on, it also needs additional components, but at least here we're saying it does not need a primer. Synthesis of RNA is in the same direction as DNA, from the 5 to 3, because these nucleotides are always loaded on, the 5' end. Sorry, the nucleotide, the incoming nucleotide is loaded from its 3' end. Okay. Now, if this is clear, and of course, if you don't understand more, okay, maybe I just missed out something here. I've already explained what the promoter sequence is, and I'll explain the promoter in detail as we continue. And I remember saying that the promoter is just a region or a segment that is found, within the gene region that tells this process of transcription, or in this case, the polymerase to say, this is where you should start reading the gene that you want to transcribe. So how does RNA polymerase synthesize RNA from the template strand? So this time around, we're no longer looking at DNA as two strands. We're looking at RNA transcription or synthesis by looking at it from only getting information from a single strand of DNA. So I mentioned to say this process is stepwise. It has to initiate, and initiation means that the polymerase must know where the gene starts, and this one is marked by this promoter. And then also it has to elongate. In elongation, what are we doing? We're adding nucleotides to synthesize our mRNA through complementary base pairing. And then the process has to terminate. Termination means that we've come to the end of the instructions or the end of the gene that the cell needs. And in eukaryotes, we're also going to go further by looking at other processes that take place once RNA is made, such as polyadenylation, like I said, and also removing those introns. Like I said, our genes have both exons and introns. The introns need to be removed for us to make a mature mRNA molecule, or should I say, an RNA transcript. Okay, this is just something that we have to think about. How does DNA itself provide instructions for where transcription should start? That is to initiate, elongate, and to stop. So these instructions, like I said, are embedded within the DNA itself, and collectively they're referred to as regulatory elements. What we mean by regulatory element is just to say they must guide the process to know where it must start, elongate, and stop. These regulatory elements include things such as the promoter and the terminator. And this guides this enzyme RNA polymerase to know where it should load, as this is where the gene starts, because if these regulatory elements are not there, RNA polymerase maybe can start reading the instructions from the middle of the gene, and some of the information is going to be left out. To ensure that the RNA product we're getting has got all the instructions within the gene, it must be guided where to start and also where that information ends. So, guiding the RNA polymerase from start to the stop of the gene, that is from the beginning of the gene to where it ends. So, the RNA polymerase performs transcription in a stepwise manner. It needs to start, elongate, and then terminate. Now, if I want you to get a snapshot of this, because I'm going to start by looking only at the promoter, then we're going to go to elongation, and finally termination. So we're going to start with the promoter. How does the promoter look like? So what you see here is a segment of DNA. In this segment of DNA, we'll start by looking at the orientation. We have a 5' here and a 3' here. Because these are prokaryotes, we're assuming that this is a circle, it's a loop with the other genes on that other end, but this time around, we've only picked a segment we're calling a gene. So a gene should have a marker, and this marker tells RNA polymerase to say this is where this gene starts. So one of the markers, or should I say the marker that guides RNA polymerase to say this is where the gene starts, is referred to as the promoter. We're going to look at the sequences in the promoter, but for now, the understanding here is that every gene, sometimes not every gene, but should I say in this case, these genes have a signal or a region that guides the polymerase where the beginning of transcription should start. This region is referred to as the promoter. And also, downstream, when I say downstream, I'm referring to sequences this side. Upstream, I'm referring to sequences this side. You can see that at this position, you have a +1. A +1, sometimes also referred to as a transcription start site, is that exact point on the gene where transcription is going to start. and mostly it reads the first nucleotide of your gene. What this means is that the promoter region, it also has nucleotides. This is just information that guides the RNA polymerase to bind. But when it binds there, this information in the promoter, it will not find itself in the final product, which is your mRNA and the protein. No. But the transcription start site is that information that is going to be part of your mRNA product. That position on your gene or segment of DNA that marks the first position of transcription is referred to as a transcription start site, or in this, you can just simply say TSS. This region is designated as +1, +1. So meaning that all nucleotides going this way are given a positive sign, +2, +3, +4, depending on the position of that nucleotide. And all nucleotides going this way are designated with a negative sign. I have a slide where I'll explain that in detail. But for this slide, the take-home message is that the gene, the segment of DNA that is carrying the information that the cell needs specifically, has got signals that allows transcription to be guided, to know that this is where this information should start and this is where this information should end. The start of this information has what is referred to as a promoter. The promoter is that region that will bind our RNA polymerase. And our RNA polymerase, when it is stable, it is now going to transcribe our gene. The region of the promoter or the nucleotides in the promoter do not find themselves in the mature mRNA or protein. This information is just necessary for identification of the start of the gene. So now, what does the promoter have? The promoter is that sequence or region on your gene that contains information for guiding your polymerase. In bacteria, it has what is referred to as a -35 sequence. This is a location where RNA polymerase binds first. It also has a sequence that is designated as a -10. Sometimes it is referred to as a preb now box. And this is where local unwinding of the DNA takes place. Remember, we said not both of the strands are going to be used in DNA, in RNA synthesis. Only one strand is used. What that means is that the process of transcription needs a single strand. The preb now box is that region of the promoter that is going to allow for local unwinding. So now let's look at this in a picture form. Now, maybe this is what you have in your notes. Allow me to use a simple diagram. It's the same thing, by the way. This is the region of the promoter. This is your +1 site, and this is where transcription is going to start. But I feel like there's too much going on here. So let me use a simpler diagram. Okay, so what you have here is a promoter region. You have these two strands. This is a DNA molecule. It's got a 5' and the 3'. And also the numbering, remember I said this is a TSS, sorry, the transcription start site. I'll just say TSS just to mean a transcription start site. The transcription start site is the position on your DNA molecule. The nucleotide at this position will be transcribed into your mRNA. This is where the information from the +1 site will find itself in your mRNA. This other information upstream of the +1 is just to guide the RNA polymerase, ensuring that it is stable and everything that is required for transcription has loaded onto the DNA. And also to ensure that part of this region unwinds because only one of the two strands needs to be used as a template. Now, in terms of numbering, like I said, we have the +1 site, which is the start of transcription. The regions that are found, I hope you can see my cursor. On this region are referred to as upstream regions. Regions that are found this way of +1, these are downstream regions. The upstream regions are designated minus. So what this means is that if this is minus 10, from this plus 1 to there must be about 10 nucleotides before this guy can start. And from here to there, about minus 35, meaning from here you must count minus 31 bases that move 35 times. You're going to find this sequence. Now, the promoter in prokaryotes has got a minus 35 consensus sequence and a minus 10 consensus sequence. What does the word consensus mean? At this point, my understanding is that when we look at a gene, or the promoter region in this case, all this yellow thing here is a promoter. And we're trying to say it has got a minus 10 and a minus 35. Where are these negative signs coming from? The reference point is the TSS, which is designated as plus 1. This is going to be your first nucleotide in your RNA molecule. Regions that are found upstream, these are called upstream sequences. And these ones are given the negative sign. What minus 10 means is when you stand here, there must be about 10 ribonucleotides here to get to this guy, which we refer to as the prep now box, right? And then to move from this to there, you must count about, it's approximately 35 nucleotides upstream. But because these regions are this way, they're given a negative sign. Regions or nucleotides going this way are designated with a positive sign. So let's say, for example, there's an adenine here at position 15. I'll say this is plus 15. What does plus 15 mean? Meaning when I start from here, I must count 15 nucleotides. 1, 2, 3, 4, up to 15. If I say the adenine is is at plus 15, what that means is that starting from here, I'll be counting 15 times to that adenine. It must be at position 15. Now, I was trying to make us understand, what does consensus mean? When you think of consensus, we're trying to say it is a sequence that is most common or highly conserved nucleotide, or highly conserved sequence of nucleotides that are found in many similar DNA sequences when they're compared. What this means is that if you get bacteria strains that cause tuberculosis, okay, you get their secular genomes and then you read them. On this gene, every time you have a plus 1, the most common sequences around minus 10 region will be the prep now. And again, when you move from the plus 1 on those genes, again, you're going to find similar sequences, T, T, G, you know what I mean? This means that this sequence is common in most genes. So because it is common in most genes, it is referred to as consensus. So consensus just means a sequence, meaning that it's got more than one nucleotide. That is common in many genes. Or in this case, it is common in the promoter regions where these RNA polymerase bind. When we say consensus, what we're trying to say, it is just a region, in this case, in the promoter, that is quite common in many genes. Where this RNA polymerase plus 1, when you move 10 nucleotides, you mostly find the prep now box. This combination of nucleotides is common in most genes at position 10. And again, the minus 35, in most genes, when we align them, we put a lot of sequences. When we align the plus 1, when we start moving this way, when we count minus 35, we'll probably find these sequences. Because they're common in more than just one gene, they're referred to as a consensus sequence. Consensus sequence has got more than one nucleotide in this case. So I hope that we have understood the structure of the promoter. In addition to that, we've also understood the way we are numbering them. When you say plus 1, what do you mean? When you say these sequences are downstream, what is your reference point? It is at this position at the TSS. These regions are designated positive. These upstream regions, upstream means they're going this way. These are indicated with a negative sign. You are going to realize there's no 0. 0 means there's nothing. But remember, all over here, there are other nucleotides. So this cannot be a 0, like 0 then minus 1. There's no 0. It is just plus 1. The next one should be minus 1, minus 2, minus 3, up to minus 10, and so on and so forth. And this region that is found upstream of the TSS is what we're referring to as a promoter. The promoter region is the region it's a segment of DNA that is found on the gene or upstream of a gene that contains a marker or an address where RNA polymerase is going to bind. It also has a region that can allow for unwinding of DNA. And what you're going to see here is that it is got a high combination or a higher value or combination of A's and T's. And we know why that is so. Because the cell doesn't want to use so much energy breaking hydrogen bonds. Breaking 3 if you had G's and C's. While in this case you've got T's and A's, it's easy for you to break 2 hydrogen bonds compared to 3. So it makes sense why this guy here, the Prebnal box, has got more A's and T's. For more notes on this, I think you can read this. If you still don't understand how to number a gene and things like that, I think we can look at it later. But at this point, I want us to move on. So this is, on this slide, we're also just trying to say the genes in prokaryotes have got promoters. Our genes also have promoter sites. They also serve a similar role. They act as an address where these polymerases, RNA polymerases, know that this is where the gene starts. The numbering of the genes is the same. We also have a TSS at +1, and upstream sequences are designated with a plus sign, and negative sequences also are designated with a negative sign. In our case, we have what is referred to as a Tata box, and this Tata box, you find it approximately 35 and another region at -35, meaning they are this way, plus they are on the other end. The take-home message here is that transcription needs a guide to know where it must start. In both prokaryotic genes and eukaryotic genes, we need these signals that mark where the genes start. And these regions are referred to as the promoter. In bacteria, we have the Prebnal box at -10, approximately at -10. We also have the 35S consensus sequence found upstream of the +1. Now, the promoter is not the only signal that transcription needs to guide where to start. It also needs a guide to know where it must stop. So when you look at this gene or this DNA sequence, you have the promoter, and we know right now why the promoter is needed. And then, of course, we know if transcription starts, it should start here, and it's going to read this strand all the way. But remember, in transcription, we're not transcribing the entire DNA molecule. At this point, we're all very clear. We're only transcribing a segment of this DNA that is carrying the instruction that we need. So in this case, for example, we have just had very nice sugar, and now we've got too much sugar in our system. We need insulin. The RNA polymerase needs to know how far does this information containing insulin end. So meaning that transcription needs a termination signal. And these genes have a region that is referred to as the terminator. And this is where transcription will end. It is a marker to say this is where your plot ends, this is where this information ends, like a beacon. This is where transcription must end. It is found downstream. When I said downstream, the reference point was a +1. Upstream of the gene, the reference point is still the +1. But this whole region here is referred to as a gene. So meaning that a gene has got features of instructions for where the gene starts, also instructions for where the gene ends. Then also this gene also has information for now making the specific instruction that the cell code for. What we should keep in mind is that there are certain genes that are produced every time. These are housekeeping genes, but this time around, think of it like this. These genes are being asked for by the cell to say, this time around, the body needs to run because there's a monster in the room. So they're going to make a call for the gene for adrenaline. Or this time around, you've had too much protein. Okay, we need pepsin or pepsinogen to be synthesized. So we don't want to be synthesizing pepsinogen anywhere, even if there's no protein in our system or amylase. When there's no start to digest, it is not digesting our own system, so we don't need that. So these genes, the expression is regulated. And what I mean by expression, guys, is that right now, the moment you retrieve information from DNA, you've started the process of expressing it. Okay, turning it into some information, that's what I mean. Okay, So now let's move on now to the process of transcription. Transcription starts with initiation. Okay, transcription is going to start with the process of initiation, similar to what we see in DNA replication. This is just an overview of what's going to happen. I'm going to go through the slide. So in transcription, we know that the enzyme that is going to be used is RNA polymerase. Please do not confuse RNA polymerase to DNA polymerase. This one is RNA polymerase because it is using ribonucleotides and not deoxyribonucleotides. The process involves unwinding of DNA. Why do we need to unwind DNA? Because we understand at this point to say we only need one of the two strands to serve as a template. Then, of course, we need to synthesize our RNA. When synthesizing our RNA, we are following the rules of complementary base pairing, and we also know the polymerase will be guided to the promoter to know where it should first bind. When we look at eukaryotes, because our genes have got a different structure, in this case, and what I mean by that is that it is interrupted by introns. The RNA transcript that is made is not yet mature. It undergoes further processing. Okay. In both eukaryotes, in eukaryotes, we're going to have the process of 5' capping and also polyadenylation and then splicing. Splicing is a process we're going to now take out those introns. Now, the reason why our RNA is further processed is because of other processes that are required. Why do we need a 5' cap? Remember, it all goes to the structure of our cells. This process we said is taking place in the nucleus. But the process that makes proteins takes place in the cytoplasm. Because transcription is not linked to the process in the cytoplasm, we need to protect our RNA. So that's why we have a 5' cap and poly A tail. I'm going to explain this further, but this one is just to remind us to say, when we looked at the slide, to answer the question, what is the site of transcription, it also helps us to understand why our RNA is capped on both ends with a 5' cap and on the other end a poly A tail, because the site of transcription and translation in the cytoplasm are different because of that compartmentalization. Okay, so this is very clear. The region that we're going to be transcribing is the green patch. I don't want to explain this further, and I hope we have understood. I'm also going to skip this slide because at this point we understand that the beginning of transcription, which is initiation, is taking place at the promoter. So the next slide I'm going to focus on is going to look at the promoter region. Understanding this is upstream of the +1, and it's got that -10 and -35 region. Okay, so let's look at the process of initiation. We know the enzyme RNA polymerase can initiate polymerization of RNA without needing a primer, but it needs something to aid it, recognize the promoter. What we're trying to say here is that RNA polymerase can start polymerization without the need of a primer, but it needs an additional, helper to help it recognize the site of the promoter. So, to recognize the promoter, and the region that is being recognized first is a -35, like I said, RNA polymerase needs the sigma factor. So the sigma factor, this is just a protein that is a component of the RNA polymerase that helps the enzyme to recognize and bind to the promoter sequence. Which part of the promoter is it binding to? Of course, it's going to bind to the -35 and then finally start moving towards the -10, where we're going to have local unwinding. So what we're trying to say here is that RNA polymerase utilizes the sigma factor. The sigma factor is a protein. This protein enables RNA polymerase to identify the promoter region and also help it bind to the promoter. It will first recognize the -35, bind, and then start moving towards the -10. Now, the sigma factor is only required for recognition of the promoter. What this means is that at some point it's going to fall off. It's going to be let go by the polymerase, because after the polymerase has recognized the promoter, there is no need for it to continue carrying the sigma factor. The enzyme is going to be heavy unnecessarily and move slow. To allow for transcription to take place efficiently, the enzyme lets go of some of its weight, those proteins that are no longer necessary. So here we must understand the polymerase to initiate synthesis cannot do it on its own, but it is aided by what is referred to as a sigma factor, which is a component of the same enzyme. And this component of the enzyme, the sigma factor, allows the polymerase, RNA polymerase, to recognize, like identify the address to say this is where this gene starts, where in the promoter, the minus 35, and then also to bind the promoter sequence and thereby initiating transcription. So now, what we're trying to say here is that this region that the RNA polymerase is binding to, just a reminder, is a promoter. The promoter is identified by the sigma factor. We can also look at promoters in eukaryotes. Now, the eukaryotic system, like I said, also has promoter regions. And of course, we won't go so much into detail, but we also have similar proteins that bind to the promoter region to aid the polymerase to synthesize. And these are not sigma factors, they're other factors, but we won't go so much into detail. But just to say that, similar to our promoters in prokaryotes, ours also need to be identified. Okay, so at this point, once the sigma factor, like I said, has identified the promoter region, and also there's local unwinding of the prep now box, which is a -10, the sigma factor is released. So what normally happens is that there must be a transition from initiation to elongation. Initiation involves recognition of the promoter and binding of the polymerase to the promoter. This recognition of the promoter and also loading of this enzyme to the promoter is aided, among other things, by the sigma factor. And once a short strand has been read of that promoter region, the sigma factor is not needed anymore. And this is because the enzyme is going to be heavy. And also the sigma factor has already served its role. It has allowed the polymerase to know where the gene starts. So the sigma factor is released by the enzyme. And when the sigma factor is released from the enzyme, it marks the entry into elongation. And the enzyme can proceed to synthesize our mRNA molecule. Of course, what you're seeing here is the whole process of transcription, but it is important for us to understand that the sigma factor aids in the recognition of the promoter and the binding of the polymerase to transition from initiation to elongation. And we know what elongation is. We're now going to add new nucleotides using complementary base pairing. The sigma factor needs to be released. The enzyme will be too heavy unnecessarily and slow down the process of transcription. Only one of the two strands will serve as the template. In addition to that, I also want us to just keep in mind that only a short segment of RNA is going to be complementary bound to the DNA. The moment it is synthesized, most of it peels off, but a good amount of nucleotides must remain bound to ensure that it does not peel off. It only peels off completely when it gets to the termination signal. Also, I also want to mention that the RNA sigma factor, once it is released, it can be reused by another RNA polymerase, meaning that you can have an RNA polymerase binding on the same gene to ensure that you've got multiple transcripts being transcribed at the same time. I want to end here so that we can I think we'll begin at we can start from here, maybe looking at this process in more detail, elongation and then termination. So I think the notes have already been shared, and we can have a good 5 minutes. I've got a lecture at 12. Is it 13 hours? So I can give you about 5 minutes. If there are any questions, I can address them now, or we can call it a day. I can see that most people have left. So what to do if you have a question, just unmute yourself and you can go ahead. I can see there's a hand.
Yes, madam. I want to ask, I didn't get the point. Why are the negative 10 and negative 15 sequences referred to as the upstream rather than the downstream?
Why are the negative 10? Okay, let me pick the one that we... Okay, yes. So, do you remember I mentioned that you need to have a reference point. And the reference point is the position on the gene, the region of DNA that is carrying the information that's going to end up in your mRNA, where that information starts. That first nucleotide on that information that's going to end up in your mRNA, that nucleotide is referred to as +1, like the first nucleotide, given a plus sign. And all the sequences that are going to go after that are going to be designated with a plus sign. And then the sequences, the nucleotides, should I say not sequences, the nucleotides that are going to come after this first nucleotide are going to be designated a negative sign. For example, if at this position I have an adenine, that adenine is going to be designated as +1. If the next nucleotide going this way, I have, let's say, a thymine, because this is DNA, that thymine is going to be given the position -1, and all other nucleotides going that way. So the reference point is this first nucleotide that finds itself in your mRNA in your RNA that you're making. All these sequences here are plus, 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 or given a positive sign. So that is how the plus and negative comes about, is to understand the orientation of the gene. Is that trying to say we have a 5' strand and a 5' time. You need to understand the orientation of a gene. When we say plus, or should I say -10, -10, it means, first of all, the region I'm referring to is not this way, which is downstream. The region I'm referring to, I need to go by the +1 and start paying attention to the sequence, to the nucleotides, this direction. If they say at position -10, there's an adenine there, I'll count 10 nucleotides. I should find an adenine at -10, meaning I'm going this side 10 steps or 10 nucleotides this way. Is that clear?
Sure, madam. Thank you.
You're welcome. Okay, guys, I think we can call it a day. We've got class tomorrow at 8 hours. I hope we can go for, I'm not sure how far we've covered, how many slides we've covered in this 1 hour, 30 minutes, but I hope we can do another 1 hour, even 20 minutes tomorrow, just to cover up for yesterday's lecture, because we are celebrating our African Freedom Day. Otherwise, have a good day, guys, and take care. We'll see you tomorrow. Bye.